toll-like receptors, knot-like receptors, rig-like receptors, and interference response. Role of nurses based on a Christian worldview. From a Christian perspective, nurses are called to serve others with humility, kindness, and love, following Christ's example as the ultimate healer. They see each patient as a unique individual made in the image of God, needing dignity, respect, and comprehensive care that addresses the body, mind, and soul. Nursing becomes more than just a job. It is a ministry of service dedicated to providing comfort, hope, and healing to people in need. Scripture emphasizes the importance of serving others selflessly, as seen in Philippians 2, verse 3 and 4, which encourages believers to value others above yourself. This perspective motivates nurses to view each patient as made in the image of God, deserving of dignity, respect, and empathy. Genesis 1, verse 27. Their work embodies the concept that caring for others is both a professional obligation and a spiritual responsibility to love others as well.
Okay, so the first thing is the danger-associated molecular patterns. So what they are molecules released by damaged or dying cells, and they signal to the immune system that there has been cell damage. Usually inside of a healthy cell, they would be inside. But once the cell dies or there's major damage, the DAMPs will enter the tissue and the bloodstream. Some examples of DAMPs are going to be ATP, NDA, RNA, and uric acid crystals. The immune system, it's important for the immune system because they are recognized by pattern recognition receptors, which we'll talk about in later slides, such as TLRs or NLRDs. And then when detected, they trigger inflammation to clean up the damaged cells and then start tissue repair. So it's really important for the immune system. And then, as I mentioned before, we have toll-like receptors. So these are proteins found on and inside immune cells. The cell surface TLRs detect bacteria outside the cell, and then TLRs inside the cell will detect viruses on bacterial DNA or RNA. They recognize pathogen-associated molecular patterns, or DAMPs. They also recognize molecules from bacteria and viruses, all to trigger an immune response. Some examples of these are TLR2, 3, and then TLR4, which recognizes bacteria LPS from gram-negative bacteria. All right. So first we have key processes of the toll-like receptors. And the first step of this is ligand binding. The ligand binding occurs, and the toll-like receptors are located on the immune cells. The main job of these toll-like receptors is to recognize and bind to the specific molecular patterns that are commonly found on pathogens, such as bacterial cell walls or viral RNA. Once the ligand binds to the receptors, it triggers the adapter molecule recruitment. This means that the adapter proteins are brought to the receptor site to help pass along the signal cell inside. Then we have signal transduction. This is a cascade of events where different kinases and signaling molecules are activated one after another. These steps eventually lead to the activation of the transcription factors. And finally, in the gene expression stage, those activated transcription factors move into the nucleus of the cell, and they turn on genes responsible for the immune response. Overall, the process goes from ligand binding on the outside all the way to gene activation on the inside of the nucleus, and this results in an immune response. Then we go to the concepts of toll-like receptors. The toll-like receptors first work by recognizing the specific molecular patterns. These are unique structures found on pathogens that allow the immune system to distinguish between self and non-self. This recognition happens through ligand binding, which you can see illustrated on the left. Once the binding occurs, it triggers a signal pathway. This is a process known as signal transduction that we went through on the last slide. Through this, the intracellular signaling molecules become activated. And then lastly, the toll-like receptors help the body to initiate an appropriate immune response, as we said before. This involves the recruitment of the adapter molecule and changes in gene expression, which leads to the production of cytokines and other immune mediators. Overall, the TLRs act as the immune system's early warning sensors, and they detect danger and start the immune response. Now I'm going to talk about NOD-like receptors. NOD-like receptors are proteins that are found inside the cell cytoplasm, and they detect molecules that signal infection, cell stress, or damage. And associated with them are PAMPs, which is pathogen-associated molecular patterns, and then danger-associated molecular patterns, DAMPs. So NOD-like receptors, or NLRs, they help the immune system recognize bacteria, viruses, or just cellular danger that enters the cell. And so they're really part of the innate immune system and act as our early alarm sensors. NOD-like means that they come from the nucleotide binding oligomerization domain, or NOD, and that just helps them activate immune signaling within the cells. And so they recognize PAMPs and DAMPs from damaged cells. And examples from this would maybe be bacterial peptidoglycan fragments, ATP, or uric acid crystals. And different NLRs recognize different signals. They're found in the cytoplasm of many cell types, and they include macrophages, dendritic cells, and epithelial cells. NLRs monitor intracellular environment for danger, and they're different than toll-like receptors because they're found in the cytoplasm, whereas toll-like receptors are found on cell membranes or endosomes. And some of the key processes of NOD-like receptors, first is recognition and activation. So NLRs will detect a microbial molecule or a cell damage inside of the cytoplasm, and then they undergo structural change. This is called oligomerization, and then this activates and signals the proteins, which will then start the immune response. And then second is signal transduction. So the activated NLRs will then trigger a gene expression that will then lead to, they'll produce pro-inflammatory cytokines, chemokines, and antimicrobial peptides. And then these will help recruit immune cells and eliminate the pathogen. And then finally, the third step is inflammasome formation. So some NLRs form large protein complexes, and these are called inflammasomes. And these activate caspase-1, which converts inactive cytokines into their active form. And this all causes inflammation, which is controlled like inflammatory cell death, and this will help remove the infected cells. And then finally is the outcome of the immune response, which means like all these effects combined of NLR signaling and the inflammasome activation, which will result in a rapid inflammation of the infection site. And then this recruits immune cells to the place of infection, such as neutrophils and macrophages, and then will clear out the pathogens in the damaged tissue.
So for RIG-like receptors, first off, to start, the RIG in RIG-like receptors is an abbreviation for the retinoic acid inducible gene. RIG-like receptors, or RLRs, are pattern recognition receptors found in the cytoplasm. Found in the cytoplasmic region of cell, and they're key sensors for virus infections. They help mediate the transcriptional induction, or the production of and activation of different genes that establish an antiviral host response, like type 1 interferons. The main members of the RLRs and are RIG-like 1, melanoma differentiation associated gene 5, or MDA5, and Laboratory of Genetics and Psychology 2, which is LGP2, right? First off, what they recognize is the RG1 are sensitive to either short or single-stranded double-stranded RNA, and recognizing the viral RNA from various families like measles. And then the MDA5 is more selective in what they look for, and they look for longer RNA double strands. And then we got LGP2 is different compared to the other two. What they do, they more bind to blunted double-stranded RNA, and they're regulators for RGI or RG1 and MDA5 activity. And where these things can be located are in the cytoplasm of where they're involved in the recognition of viral RNA. So it can be found in many different cells as long as it's something that helps with fighting viral infection and things like that.
Okay. RGI-like receptors are part of our innate immune defense system. They act as sensors for viral RNA inside the cytoplasm of infected cells. So they first recognize viral RNA that looks different from the cell's own RNA. And then when the viral RNA will bind, the RGI will activate by changing shape, and it exposes its card domains. So these card regions then interact with MAVs, which is a signaling protein located on the mitochondrial membrane. This interaction is a critical step that connects viral detection to an immune response. So the MAVs will then trigger a signaling cascade involving kinase, like TBK1 and IKE, which will activate the transcription factors IRF3 and IRF7. Finally, these transcription factors move into the nucleus, which stimulate the expression of type 1 interferons and pro-inflammatory cytokines, which will alert the neighboring cells. They'll help contain a viral infection, and overall, the RLR pathway will provide a rapid and essential response to viral threats before the adaptive immune system kicks in.
So the interferon response plays a crucial role in our body's immune system, which is the first line of defense against infection. Where a virus infects a cell, that cell produces interferons and a group of signaling proteins, also known as cytokines. These interferons act as messengers, warning nearby cells of viral presence and triggering antiviral defenses to limit viral replication and spread before the adaptive immune system, which involves T and B cells. The name interferon comes from their ability to interfere with viral replication, essentially slowing down the infection. Beyond fighting viruses, interferons also regulate inflammation and enhance antigen presentation, which helps activate T cells later in the immune response. In short, the interferon response is a bridge between the innate and adaptive immune systems, ensuring the body reacts quickly and effectively in viral infections. And these interferons have functions or pathways that they use to fight these or kind of regulate these immune responses. And they have different types of classes, just two, which is type 1 and type 2 IFNs, which is short for interferons. And they both activate common and distinct STATs, which are signal transducers and activators of transcription. They regulate the transcription of target genes. In addition, the classical Janus activated kinase, or JAK, and STAT signaling pathways, both type 1 and type 2 IFNs activate several other signaling cascades. And then we got the CRKLs, a member of the CRK family of proteins, is activated by both of the IFNs. Its function is as an upstream regulator of signaling pathways and involves the guanine nucleotide exchange factor, C3G, and RPI1.
And then these are our references.